Welcome to this tutorial video. So this is part 2 of bacteria session. I just hope that you've watched the part 1 video on bacteria. So on the part 1 video, when we're looking at bacteria, we talked about the introduction to bacteriology, basically talking about the bacteria. We also talked about the shape of the bacteria. We also looked at the different structures that you find on a bacteria. For example, we looked at the flagellum. Okay, we looked at the pili, the fimbriae, we looked at the cells and everything. And then we also looked at the functions of those different structures that you find on a bacteria. Now, in today's session, we'll be looking at the classification of bacteria. It's very, very important as a medical student to understand these different classes of bacteria. Why? Because when you start learning what we call pharmacology, you find that certain drugs that we give to patients who've got bacterial infections, we give them based on the fact that we understand the shape of a certain bacteria. Maybe we understand, does this bacteria like oxygen or not? There are different things that we put into consideration as we are giving those different drugs to patients who have got bacterial infections. So before you even start learning about pharmacology, it simply means that it's a requirement for a medical personnel or a student to understand these different classifications of a bacteria. So we're saying that bacteria are classified in three main ways, and that is according to one, shape and structure. So when we're classifying bacteria, remember we said bacteria, they exist in numerous ways, in numerous numbers. They are countless. So they're in different forms. So how do we get to simplify things when we want to know which drug we must use or which approach we must use? We want to classify them. And one of the ways we can classify bacteria, remember when I said bacteria, it's plural, right? Is see according to shape and the structure. So we're going to be classifying this bacteria depending on the shape. How are they looking in terms of shape and structure? Apart from that, we can also classify bacteria based on oxygen requirement. So on this particular concept, we want to understand, does this bacteria like oxygen or not? If it doesn't like oxygen, even you as a medical personnel, if you understand, okay, if this bacteria doesn't like oxygen, it simply means that if I put it in an oxygen environment, it's going to die because it doesn't like oxygen. Then apart from that, there is this one which is done, which referred to as gram stain. This is one of the most important classifications that we use in medical sciences. So there's what we call gram staining, or we'll be talking about gram positive and gram negative bacteria. So quick enough, a recap, we're saying we're looking at the classification of bacteria. We're saying that bacteria, broadly, they can be classified according to shape and structure, oxygen requirement, as well as gram stain. So we're going to go through each and every class so that you understand, if we're talking about classifying a bacteria based on structure, then how are we classifying them? Remember, this is important in in pharmacology. When you are choosing specific drugs that must be used against a specific disease or infection. So we're going to start with the first class, which is classifying bacteria according to shape. Okay? So for the students who are in school, like maybe for the BScs, the RNs and everything, you are required to have an idea of the people that were involved in classifying these different bacteria. Okay? So in the year 1872, a scientist known as Ferdinand Cohn, okay, classified bacteria to major four types depending on their shapes as follows. So when you are classifying bacteria depending on the shape, on this particular concept, you are going to classify bacteria in terms of cocci. So whenever you see a bacteria with the name of cocci, streptococci, it's telling you that this kind of bacteria has got a spherical shape. I just gave an example of streptococci bacteria as an example. I'm sure some of you have come across that bacteria, that name before. So whenever you hear or you see the cocci, term, okay, in the name of a bacteria, it's telling you that this bacteria has got a spherical shape or round shape. They are also what we call vibrio. I think some of you have heard of what we call cholera disease. And one of the major cause of cholera disease is what we call cholera vibrio bacteria. So they are also what we call vibrio, bacteria. I'm going to show you how they look. They appear more like a comma, the same comma that you use when you're writing statements. Okay? They are also what we call bacillus, okay, or bacilli bacteria. These ones are rod-shaped. When I say rod shape, they appear like a string. For example, some of you have heard of what you call tuberculosis. It's a disease that is caused by what you call mycobacterium, tobacco, bacteria. So that one is a rod-shaped bacteria. No wonder it has got the name what we call bacilli. So apart from that, there are also what we call spirilla. So spirilla is coming from the word spiral. Okay, I'll show you how these structures look. What I want you to know here is that when you are classifying bacteria based on the shape, you do it in 4 different ways. One, cocci or spherical, meaning this type of bacteria, they are spherical. So when you hear pneumococci, streptococci, staphylococci, it's telling you that this kind of bacteria has got a spherical shape. Okay, the vibro ones I'm going to show you, the bacilli one. These are rod-shaped bacteria. They appear like a rod. If some of you have seen sausage before, they appear like that. Okay, they are also what we call spirilla bacteria. So this picture that you are seeing is what I'm talking about. We're going to start with the cocci. So cocci, we say these ones are spherical. They are spherical. So for example, when you hear a bacteria being named as strep, a strep is more like a chain. I don't know if some of you have seen a chain before. So we've got, since we said cocci means spherical or round, it simply means that you've got a chain of round things. So this kind of a bacteria is going to appear like a chain of connected round balls, like you can see here. So they will be referred to as a strep. Okay, strep. Okay, some of these, maybe some of you have heard of what we call strep throat. They are called by streptococcus bacteria. Apart from that, they are also what we call staphy. Staphylococcus aureus, maybe some of you have heard of that kind of bacteria before. Okay, it causes infections such as sinus infections. You're going to be learning about these things when you start learning about medicine and the stuffs. But what I want you to know is that when you're classifying bacteria based on the cocci or spherical shape, there are 2 things you have to put into consideration. When you hear strep is like a chain. When you hear staphy, I don't know if some of you have seen what we call, there are these fruits which we call Maulbeer fruits. The way they appear, they are clustered together. So you've got a lot of circles that are clustered together like this. We call them staph. So in microbiology, we're saying this is a staphylococcus kind of bacteria. So that is on the spherical or cocci. So if they use the word spherical or cocci, that is one and the same. Then I also said that they are also what we call rod-shaped bacteria. These ones, we call them bacilli. So these ones are rod-shaped. They appear like this, like a rod. Okay? Rod-shaped bacteria. For example, Bacillus anthracis. This kind of bacteria, you find it causes, disease in animals, like maybe the cattle and everything. Okay? So these, they appear like sausage, like as you can see, they're rod-like. Okay? So these ones are rod-shaped or bacilli. For example, Clostridium, Bacterial bacteria causes what we call botulism. So botulism is a kind of disorder that affects the nervous system. Okay? I'm just giving an example. What I want you to know on this element, on this part, is that rod-shaped bacteria appear like a rod. Okay? No wonder we call them bacilli. Apart from that, I said they are what we call spiral-shaped bacteria. Okay? They are not straight in terms of their structure. It's kind of meandering in terms of their structure. So you've heard of what you call Vibrio cholerae. This is the bacteria that causes cholera. It falls under what we call the spiral-shaped bacteria. Some of you have heard of what you call Helicobacter pylori. Helicobacter pylori is the leading cause of what we call peptic ulcers. If you start learning about surgical nursing and the stuffs, you look at peptic ulcers. So Helicobacter pylori is one of the major causes or leading cause of what we call peptic ulcers. We're going to learn about it. It has got a spiral-shaped structure. Okay? Some of you have also heard of a disease or a disease that we call syphilis. Syphilis disease is very common. It's an STI. It's caused by what we call Treponema pallidum bacteria. This one is also a spiral-shaped bacteria. Remember I said in the beginning that the reason why you are learning about these different classes of bacteria is that when you want to intervene in terms of pharmacotherapy or giving drugs to a patient, okay, you put into consideration the shape of the bacteria. When I'm giving this kind of drug, okay, if you start learning about pharmacodynamics, pharmacokinetics, there are things that you put into consideration that you need to relate now to microbiology as you are giving that particular drug. Otherwise, at this particular point, you are required to have this knowledge on these different shapes of bacteria. Spherical cocci, rod-shaped bacilli, spiral-shaped, these are the ones such as Vibrio cholerae, Helicobacter pylori, as well as what? Treponema pallidum bacteria. Okay, if you want more pictures on the shape, you can simply pause the video. I've put a lot of examples for you here. It's the same picture as the previous one. It's just that here I've put a lot of examples that you can use if you want to you want to familiarize yourself with some of the examples of the actual bacteria that exist in terms of the spherical one or the rod shapes as well as the spiral bacteria. Apart from that, I said, besides classifying bacteria based on the shape, we also classify bacteria based on oxygen requirement. This is also very important because you'll find that some patients, maybe they will be required to be given oxygen therapy and everything, and then you also find that the very patient is also maybe fighting an infectious agent that likes oxygen. So what do? You now start fighting, because if you know this kind of bacteria that has infected these patients likes oxygen, then these patients, for them to survive, you are supposed to put them on oxygen therapy. Then I think you will not use oxygen as a proper way of killing that microorganism. You're supposed to find another alternative. The principle or the concept is that you must know to say some bacteria, they like oxygen. Some of them, they don't. Some of them, they just like oxygen, just in a little amount and everything. So let's look at classifying bacteria according to oxygen requirements. Now, from secondary schools, I think if you've been following through, you are familiar with what we call aerobes. When you hear the term aerobes, aerobic respiration, some of you have come across that term to say aerobic respiration. We're talking about respiration where oxygen is involved, and aerobic respiration where you don't involve oxygen. So there are bacteria which we classify as aerobes. So the fact that we're saying aerobes, these bacteria need oxygen to grow, using it in their metabolic processes. So a bacteria, in as much as it is just one cell, like I've always explained, it also has to undergo certain metabolic processes for it to grow, for it to have food, for it to perform various functions that it does. So if you come across a bacteria which is being referred to as or falling under a class of what we call aerobes, it's telling you that this bacteria needs oxygen to grow. So as a medical personnel or as a student who is in the field of medicine, what is that telling you? Should you remove this bacteria from the presence of oxygen, it simply means this bacteria is going to die because it needs oxygen for it to grow. That's why you're learning these things. Apart from that, there are also what we call anaerobes. So when you say anaerobes, it simply means that these bacteria are harmed or killed by oxygen. So once you expose them to oxygen, they will die. They don't want oxygen. Okay? So what I will advise you is that for the sake of gaining more information, just go on Google, try to type examples of aerobes, okay, bacteria that like oxygen. You're going to find a lot of examples because they are numerous. Remember, we're just classifying them, we're just broadly categorizing them. So aerobes like oxygen, they need oxygen for them to grow. Anaerobes, they are harmed or killed by what? Oxygen. So anaerobes, they don't need oxygen. And then apart from that, there are also what we call obligate and aerobes. So when we say something is obligate, it simply means that it can only work in that particular direction. Okay? If we say obligate anaerobes, it simply means that these bacteria cannot tolerate oxygen at all and will die in its presence. So obligate anaerobes, these are type of bacteria that must never be exposed to what? To oxygen. Otherwise, if they do, they will die. So we call them obligate anaerobes bacteria. They just don't want oxygen. They just survive without oxygen. Apart from that, they are what we call facultative anaerobes. Okay? Facultative anaerobes. So actually, this is supposed to be facultative aerobes, not anaerobes. This is the typing error here. It's supposed to be facultative aerobes. So these bacteria can use oxygen for growth if it's available, but they can also grow in the absence of oxygen, switching to their metabolic pathways. Remember, I said this is facultative aerobes, not anaerobes. This is just a typing error. Okay? So in parasitology, you come across what we call facultative parasites. I'm just giving an example that you understand. So when they say a parasite, a parasite is a kind of organism that benefits from the host without the host benefiting, right? So it simply means that this kind of parasite will be surviving inside the host. But if they say it's facultative, it simply means that it can survive with or without the host. In the same way, we're going to use the same principle when we say facultative aerobes. It simply means that these bacteria can use oxygen, but even if oxygen is not there, they can still survive. So we're going to call them facultative. So we're saying these bacteria can use oxygen for growth if it's available, but they can also grow in the absence of oxygen. So a bacteria that can grow in the presence or absence of oxygen, we refer to it as a facultative aerobe bacteria because it can switch. If there's no oxygen, it will switch to an aerobic pathway. If there's oxygen, it's going to switch to an aerobic pathway. Apart from that, there are also what you call microaerophiles. So micro means small, aero meaning air, right? So when you say microaerophiles, it simply means that these kind of bacteria, they like oxygen but only little amount because we're saying micro. So these bacteria require low concentration of oxygen to what? To grow. So we call them microaerophiles. Okay, microaerophiles. I hope you are following. So this picture here is trying to summarize most of the content that I've talked about as you are classifying bacteria based on oxygen requirement. As you can see here, I've got this picture which has got take them as test tubes. So you've got this test tube 1, 2, 3, 4, and 5. So these test tubes, we are going to assume that we've got high concentration of oxygen on this end, and then we've got low concentration of oxygen on this end. And then these green spots that you are seeing here, consider them as bacteria. So the principle is that if this kind of bacteria, when you go to the lab, you want to do an experiment, right? If you go to the lab and then you put oxygen inside these different test tubes, and then you put bacteria inside, different type of bacteria inside. So the bacteria that is going to move towards oxygen, you are going to tell this one likes oxygen. It's going to be what? Aerobe. Okay, they're going to be aerobic bacteria. But those that are going to move away, those that are going to settle down or sediment below this particular test tube, those meaning they'll be running out from oxygen. So I'm going to tell these ones they don't like oxygen. What are you going to call them? Anaerobic bacteria. It makes sense, right? So that is the concept that we want you to understand here. So we've got test tube number 1, number 2, number 3, number 4, and number 5. So let's look at this one. We understand that the concentration of oxygen is higher towards this end and lower towards this end. So if we put or we introduce bacteria inside here, you can see that this arrow is showing that this bacteria is going towards the oxygen end, where there is a lot of oxygen. So it simply means that these are obligatory aerobes because they just want oxygen for them to survive. So they require oxygen and can't survive without it. This is why we're finding that these bacteria are going towards the high concentration of oxygen in this test tube. And then you come to these ones, facultative aerobes. So they are aerobes meaning they want oxygen, right? But the fact that they're facultative, remember I said when we use the term facultative, it simply means that they can survive in the presence of oxygen, or if oxygen is not there, they can simply switch to a non-oxygen pathway. That's why you're finding that a lot of them are towards the high oxygen concentration, but even some of them are below, they're towards the low oxygen concentration. It's telling you that these can survive with or without oxygen. So as a nursing student or as a medical personnel, it's telling you that using oxygen to kill this bacteria is not going to be a way. Because without oxygen, they will survive. Without oxygen, they're going to survive. Okay? So use oxygen but can survive without it. Then there's this third test tube here. They are what you call aerotolerant anaerobes. So the fact that they're anaerobes, remember, there's just this word aero, meaning there's air involved, right? Tolerant, meaning they can tolerate air, right? So the fact that these ones are anaerobes, they are saying that they don't use oxygen. They are anaerobes. Anything that is anaerobic does not use oxygen. So if we say aerotolerant bacteria or aerotolerant anaerobes, these they don't use oxygen, but they can grow in its presence. Not that they can use it. It's different from facultative. The facultative ones, If oxygen is there, they're going to use oxygen. If it's not there, they can still survive without oxygen. They are going to use the non-oxygen pathway. However, with the air-tolerant anaerobes, these ones they can only survive in the absence of oxygen, but they can still exist. They can't die if the oxygen is not there. Why? Because they can tolerate it, not that they can use it. That's the difference. And then there are also what you call obligate anaerobes. These ones, they just don't want oxygen. Why? Because you can see that in this test tube, the high concentration of oxygen is towards this end, the low concentration of oxygen is towards this end. They have settled down because they don't want oxygen. So we are going to call them obligate anaerobes, unable to use oxygen and can survive in its presence. No wonder they have settled down here. They are running away from high concentration of what? Oxygen here. And then last but not the least, I did mention that they are what we call microelophiles. These ones, they want oxygen, but not in high quantity, in low concentration, if you will. No wonder you find that if you look at this test tube and the direction, you can see that high concentration of oxygen is being found here. But look at where these bacteria are settling here. Meaning they are towards oxygen, but not too much of it. So I'm going to say micro, little amount of oxygen. They use oxygen, but only at a very low concentration. So I'm going to call them microelophiles. Students, this is a very, very important concept that you need to understand as you are classifying bacteria based on the oxygen requirement. You can see that from these different classes, we've been able to see that they're kind of bacteria that just want oxygen. They are kind of like for example, obligate aerobes. Then they are type of bacteria that can survive in the presence or absence of oxygen. We call them facultative aerobes. They can switch. If there's no oxygen, they'll start using an aerobic pathway. If there's oxygen, they'll start using the aerobic pathway. And then there are those which we've said, which we call aerotolerant and aerobes. These ones, they can't survive without oxygen. I mean, they can't survive in the presence of oxygen, but even if it's there, they're going to survive, but they're not going to use it. So we call them air-tolerant anaerobes. Then we've talked about obligate and aerobes. These ones, they are obligated to an anaerobic pathway. They don't need oxygen in any way. Then we've talked about the microelophiles. These, they want or they need oxygen only in small concentration. Okay? So if you've understood, try to solve this kind of puzzle, right? There's test tube A, test tube B, test tube C, test tube D, and E. I can give you leakage. Like I can answer, these are what you call microelophiles. These ones are what you call obligate aerobes. These ones definitely are going to be what? Obligate and aerobes that don't want oxygen. What about C and D? Try to answer it on your own based on the concept that I've explained. Apart from that, the last classification that I'd mentioned was you being able to classify bacteria according to what you call gram stain. So gram stain is a term that comes from the scientist that discovered this concept of classifying bacteria based on the staining principle. So of all different classification systems, the gram stain has withstood the test of time. What we mean is that among all these different classes of bacteria classification, classifying bacteria based on the shape, based on oxygen requirement, nutrients, temperature, and everything, the gram stain stands out to be the most important one and the most used one. So this one was discovered by what we call Hans Christian Gram. I've just abbreviated it in 1884. So Hans Christian Gram was the scientist that discovered on how to classify bacteria based on the gram staining principles. It remains an important and useful technique up to this particular day. So when you start doing what we call pharmacology, they are going to tell you these are gram positive bacteria, these are gram negative bacteria. So there's a difference in how these different bacteria operate. In case they ask you who discovered the gram staining principle, you tell them it's Hans Christian Gram in 1884. And this is the most important classification system among all the classes that exist around the world. So let's explore and see what he discovered. So gram positive and gram negative bacterial staining, they are different in terms of how they look. Before I even go into explaining what is involved, most of you are going to have a chance to do what we call labs. So when you do labs, they are going to show you how a gram-negative bacteria appears in terms of color and how a gram-positive bacteria appears in terms of color. So I've used these colors not because I want to decorate the notes and everything. No. These colors are what you see when you go into the labs, and then they'll ask you, see through this microscope. So the gram-positive bacteria have got what you call a bluish or purplish appearance if you view it under a microscope. Then the gram-negative bacteria have got what you call a pinkish reddish appearance if you view them under a microscope. So these are the colors that you are seeing here. So we've got gram positive, and then you also have what we call gram negative. Gram positive, gram negative. Okay? So let's also look at the other differences that you need to know as a student or as a medical personnel as you talk about gram positive and gram negative bacteria. So what makes us say these are gram-positive bacteria, or how do they appear? In case they ask you, describe gram-positive bacteria, what do you have to say? So we're saying that the gram-positive bacteria, they retain the purple color and look blue to purple because they've got a larger peptidoglycan or cell wall. Remember when I was telling you when I introduced the topic on bacteria on my first video, I said you have to pay particular attention to the shape of the bacteria because it has got direct influence on how the drugs that will be given to this particular patient, how they'll be working. So there's what we call the cell wall. Within the cell wall of a bacteria, there's what we call a peptidoglycan. It's a layer that is made up of proteins and carbohydrates. We call it peptidoglycan. Peptide coming from proteins, glycan from the glucose. It makes up what we call the cell wall. So if you are talking about the gram-positive bacteria, we're saying that when you stain them with the special, formulas, okay, or liquids that are used in the lab when you want to figure out is this a gram-positive or gram-negative bacteria. So once you stain them, or you use what we call some form of dye, you dye them, and then you view them under a microscope, you find that the gram-positive bacteria is going to retain. If we say retain, meaning if you stain it with that purple color, it will still be purple. It will not change. It's going to retain that purple color. What I was saying, the gram-positive bacteria, they retain the purple color and look blue to purple because they've got a larger peptidoglycan layer, the cell wall. So there's what we call iodine and the crystal violet precipitate in the thickened cell wall are not eluted by alcohol when stained. So when you use what we call iodine and crystal violet precipitate, these are the materials that you use when you want to stain these different microorganisms if you want to check if they're gram positive or gram negative. So once you expose them to what we call alcohol, when we say iodine and crystal violet are not eluted, it simply means that they're not removed from the cell wall. It's like you get a shirt, then you dip it in a bucket of paint. So once you do that, you find that this shirt is going to retain that color. If the bucket of paint had pink in it, that will be pink. If it will be blue, it will be blue, it will be blue. So the gram-positive bacteria, when you stain them with this kind of staining material or liquid or formula, they're going to retain the very color that you used because they've got a thick peptidoglycan layer. For example, there's what we call Corynebacterium and what we call Clostridium. These are just examples of the gram-positive bacteria. There are a lot of examples that you're going to find down here. Okay? So, looking at the structure of the gram-positive bacteria, see, these are gram-positive bacteria, and then I think I've already shown you in my first video or on what we call the cell wall. So this is the cell wall of a bacteria, right? You've just gotten a piece. This is the bacteria, right? You just get a piece of the cell wall. So this is the cell wall. So this cell wall, I said, it has got in gram-positive, it has got what we call a large peptidoglycan layer. It's thick. So this is the layer that I'm talking about. This is the cell wall, right? And then if you zoom in on this cell wall, this is the outer part which has got the thick peptidoglycan layer. Peptidoglycan because it's made up of peptides and glucose. Okay? And then it also has what we call, of course, we're not going into details talking about the plasma membrane. No. We're interested in what? The cell wall, the peptidoglycan layer. So when you stain this bacteria with the iodine and crystal violet, and then you expose it to alcohol, you find that that stain is retained because this peptidoglycan layer is thick enough to be able to retain the purple color that you use when you are staining it in the lab, hence making it gram positive. So once you just see that, You stain it, the color doesn't change, it still maintains, it's just blue. And then you view it under a microscope, you're going to say, oh, this one is a gram-positive bacteria. And just so you know, gram-positive bacteria are easy to kill as compared to gram-negative bacteria. Why? Because they have got a larger or thicker cell wall, which gives at least a reasonable surface area for drugs for them to be able to penetrate through this particular bacteria. Let's also talk about the gram-negative bacteria. So the gram-negative bacteria, they look red to pink under a gram stain. I showed you in the first place. So they have got two plasma membranes and a thin peptidoglycan which eludes. So these ones were saying that a gram-negative bacteria, it's different from a gram-positive bacteria because one, these they look pink to red to pink, right? The gram-positive was blue to purple. Gram-negative it's red to pink. The other difference is that the gram-positive bacteria, they only have what? One plasma membrane. However, the gram-negative bacteria has got what? Two plasma membranes, which eludes the color when you stain it. I'll explain what I mean when I say eludes the color when you stain it. They cause different types of infections than gram-positive bacteria. It's telling you as a medical student, as a medical personnel, that the gram-negative bacteria, they are very difficult to deal with in case they infect somebody. Because one, they've got a thin peptidoglycan, which makes it difficult for the drugs to be able to, penetrate. And then they also need different types of antibiotics to treat them. For example, Escherichia coli. I know that most of you know Escherichia coli as just a normal flora that is found, of course, in the body. Okay? They're also what we call Pseudomonas aeruginosa. These are examples of gram-negative bacteria. So the concept is that any gram-negative bacteria is going to give you difficulties in killing it. Okay? Or making it static in terms of growth. Why? Because it has got double layers. It has got double layers of what? Of the cell, right? As well as it has got a thin peptidoglycan. Okay? So it has got two layers of the plasma membrane, as you can see here. Let me show you. So this is a gram-negative bacteria. You've got a cell wall there, and then it has got two plasma membrane. It has got a plasma membrane on the outer part, then you've got a peptidoglycan layer in between these two plasma membranes. So you can see that it's very, very difficult, even for the stain that you're going to use in the lab, to be able to penetrate and enter through the what? The peptidoglycan layer, because it is only in this layer where that stain or dye is contained. As a result, you find that it's not going to retain the purple color. It is going to turn into what we call pink to red appearance. So that's what I wanted you to know on the gram-negative bacteria. In the next video, I'm going to talk about growth and development of bacteria, where I'll be talking about the bacterial growth curve, the lag phase, okay, the exponential phase, the decline phase, and everything. For now, ensure that you watch the video on the part 1, you also watch this video, such that when I come to teach or explain on the growth, because that's the last part of the bacteria, you understand and you follow through everything. Remember to subscribe so that you don't miss out on other upcoming videos.